Today we have a presentation by Flint Hughes. Hughes is a research ecologist with the U.S. Forest Service at the Institute of Pacific Islands Forestry. His presentation will be on Rapid Ōhiʻa Death, Patterns, Impacts, and Solutions. His research is conducted through a permit with the Hawaiʻi Experimental Tropical Forest. Please note that permits are required when entering the HETF for research purposes. Flint, you may start whenever you're ready.
Okay, thanks, Tristan. Thanks so much, and thanks for inviting me to give a presentation. I'm happy to do that and happy to be here. And I'm sorry, I have to say I can't see anybody on the screen the way we have the screen set up. So I'm going to be relying on Tristan to kind of handle any questions or during the presentation, but I do want to amend a little bit of what Tristan said. If you have a question about a particular portion of my talk that I'm on at the time, please do feel free, if you like, if it makes sense to you, to ask that question right when we're in that particular part of the talk. I'm happy to take a question. So just let Tristan know and then unmute and then ask me the question. But like I said, I can't see anybody, so I feel a little blind in terms of interacting with folks. But so good. So yeah, I like I said, I'll be talking about Laupohoihoi a fair bit, but in the context of Rapid Ōhiʻa Death, and I do want to take a little bit of time during the talk to go ahead and give some more general information that's relevant to what we're learning about in Laupohoihoi, but also relevant to Rapid Ōhiʻa Death across the rest of Big Island as well. So yeah, with that, I just want to acknowledge some of the folks that have contributed to the talk, to this talk that I'm going to give today, and are listed as my co-authors. So Kyson Dunn, Joseph Rocco, Brian Tucker, and Kealoha Kinney. And Kealoha in particular has provided some great information about the presence of Ceratocystis inoculum on feral ungulates. So I'll be getting to that in a little bit. I just want to acknowledge his contribution, which has been really instrumental to what we're learning. So let's see. Tristan, I don't know if you can hear me, but for some reason I can't advance the slide. Can you advance the slide?
Okay, just a second. Let me try for you, but I think I can't from here. Just a second.
Can you try click on the screen, Flint, instead of using the arrow, or just maybe just re-click onto the, there we go.
Yeah.
There we go.
Okay, now, okay, it was frozen for some
All
reason.
good.
Okay, perfect. Thanks, Olivia. Thanks, Tristan. So yeah, so I just want to first acknowledge the folks who have done a lot of the work that I'll be talking about today. And this is the newest version, the 2023 version of the ROD Squad, and that's constituted by Kyson Dunn, Joseph Rocco, and Sienna Dahlg, who's our newest member. And so I am so happy, privileged, grateful to work with them. They're all super individuals and they make for a fantastic research team. Also want to thank folks who have helped in collecting some of the data and the findings that I'll be presenting today. They're not working with us so much anymore. Gabby Benito and Joseph Marshall in particular were really instrumental in setting up a lot of the work that we have done. And then Brian Tucker is still working with us and hopefully will continue to do so. He has been instrumental in all of the data management of all of our research findings that we have collected over the years that deal with Rapid Ōhiʻa Death. So big mahalo to all of them, and especially to Brian, who's we couldn't be doing a lot of the work without his assistance. So just a little background about, you know, why we are so concerned about Rapid Ōhiʻa Death. And it really boils down to just how important ʻŌhiʻa is as a tree species in Hawaii. And I imagine most of the folks on this call or on this talk understand that. You know, it's the most, ʻŌhiʻa is the most abundant and widespread native tree species in Hawaii. It's been in Hawaii for at least 4 million years. And during that time, it's spread across all of the islands, pretty much establishing itself and becoming the keystone species in just about every environmental condition across the islands, whether it's humid, wet forests in the lowlands of Big Island or up in the cool bogs of Kauai, wet forest, dry forest, montane forest, lowland forest, it's everywhere. And it constitutes, as I said, the most abundant native tree species. One thing this map shows quite well too is that ʻŌhiʻa really constitutes our primary watersheds across all of the islands, really. It is the key component in the forests that are capturing our water that's available for all the organisms across the islands and also for humans. It's really providing us with our fresh, abundant, clean drinking water. And so any threat to ʻŌhiʻa is kind of a threat to all of us, frankly. In addition to its watershed value, it also constitutes a huge amount of our above ground forest carbon mass across the islands. It's by far the most massive, literally speaking, massive tree species across the island, and it constitutes the most massive forest, native forest at least. And that's why when we start to see extensive mortality of ʻŌhiʻa across landscapes due to Ceratocystis induced Rapid Ōhiʻa Death, that's a big concern, because we know how important ʻŌhiʻa is to each and every one of us across the islands. Just focusing in on Lapahoihoi and Puvawa, just a bit Puvawa as well. These are some images that just show you the distribution of forest carbon, above ground forest carbon across the 2 experimental units of HETF, the Hawaiʻi Experimental Tropical Forest. The image on the left shows the boundaries and the distribution of carbon across Laupohoihoi. On the right is Puvawa. And I just want to point out, if you look on the image, you'll see a little blue A. That's really kind of the heart of ʻŌhiʻa country, of ʻŌhiʻa forest country on the island. That's the area that's commonly referred to as the Valley of the Giants, and extending from there, that's dominated by ʻŌhiʻa forests, along with its associated other species of plants and animals. And that particular forest constitutes the largest amount of carbon mass in any native forest across Hawaii. And those forests are comparable to any, in size, in carbon mass, to any neotropical, tropical rainforest, wet forests across the entire world. They're the same size. They're obviously have different levels of diversity, but those ʻŌhiʻa forests constitute every bit as big carbon mass as any other forest, any of the large tropical forests across the world. And that's due to a couple of things. First, ʻŌhiʻa as a tree has very dense wood. And so each tree contains quite a bit amount of carbon mass. So it's a heavy tree. The density of trees across a forest stand, particularly this one, tends to be quite high. So you've got these big trees, they're quite dense as individuals, and then the stands themselves for big trees are quite dense as well. So that's the reason why these forests are so massive in terms of carbon mass. And so again, any threat to these forests is quite important and quite concerning. And so that's why we're concerned about rapid ʻŌhiʻa death in these particular areas, kind of the heartland of ʻŌhiʻa. So this is not Laupohoʻi. This is an area up above, but I just wanted to give you a feel for some of the amount of death, mortality that Ceratocystis, the fungal pathogen Ceratocystis, can cause. This is actually a forest on Mauna Kea, upslope and a little bit towards Hilo from Laupohoʻi. And it's striking, it's sad to see that, and this has occurred well within the last 10 years, probably within the last 5 years, to see this, and this mortality that is close to 90% in terms of the number of trees that have died in the area. And so this is the kind of mortality that we are really concerned about, because it's an essentially devastation of these forests. This is some island-wide scale work that Nick Vaughn and Greg Asner have done, mapping ʻŌhiʻa mortality from rapid ʻŌhiʻa death over the years. And this is a snapshot of 2017, I believe. Yeah, 2017, showing the recent mortality across the areas. And I'm showing you all this just to give you a feel for the widespread nature of the mortality. This isn't a comprehensive image. This image is not comprehensive of the mortality of ʻŌhiʻa, but it gives you a good feel about the distribution of mortality across the island, where it is, which is unfortunately it's occurring in most of our ʻŌhiʻa forests across the island, to varying degrees, of course, but it's fairly widespread at this point. Focusing in on Laupohoʻi, this is a map of fairly recent mortality of ʻŌhiʻa across that area that I was just showing you in the forest carbon map. So this is very close to the Valley of the Giants. The area defined by that pinkish boundary is really the Valley of the Giants, in specifically speaking. And what you can see is that mortality is quite high down below. The Mackay side of the image is this upper right corner, and the Mauka side is the lower left corner. So the white dots represent trees that have died a fair bit ago. They don't have leaves on them anymore, or if they have leaves, the leaves are quite silvery, and the red dots constitute much more recent mortality that are showing up as brown crowns across the landscapes. So that gives you an idea of the extent of the mortality that we're seeing in lukuohia right now, which is alarming. This is very recent. This image is from January of this year. And we did not see this level of mortality in the forest until fairly recently, maybe within the last 2 years. Before that, we had an outbreak of mortality, a very small one along Blair Road in Laupohoihoi, but it was only a handful of trees. Down farther towards the ocean, there was quite a bit of mortality that had erupted. And we suspect that mortality from Mackay, from down below, probably migrated up into the Laupohoihoi Forest Reserve and Natural Area Reserve, but fairly recently. Unfortunately, what we think we're seeing here is a movement of mortality upslope, where you see upslope is clearly the mortality is not as thick. But we think we're seeing the movement going up for reasons that I'll explain in a little bit. Just a little background about what we've been seeing through our network of mortality plots that are across the island. On average, looking at our plots within the mortality network, where we're inventorying them every year and we're developing a mean annual rate of mortality across the plots and as individuals and as a whole, what we see in general is an average mortality, average annual mortality in plots where lukuohia is present. That average mortality is about 10%. Which may not seem like a lot, but actually what it constitutes is if a given stand is experiencing 10% mortality year after year after year after year, that stand will be completely dead in a little less than 20 years time. So 10% is a significant amount of mortality. Now, that's average mortality. There are many plots that have much lower mortality rates than 10%, and we see in the associated orange and gray bars. Those constitute plots that either don't have lukuohia, they only have huliohia, the less virulent strain of Ceratocystis, or we're not seeing any Ceratocystis species in any of the trees that we're sampling. Those levels of mortality in those plots are much smaller than what we're seeing with lukuohia, which is by far the more virulent of the two species in terms of killing ohia trees. As I said, average rate, many plots that we are sampling have much lower rates than that, even when they do have lukuohia. That's based on environmental conditions, stand age, management, and management conditions, which I'll get into in a little bit. But you can see we do have plots that have incredibly high amounts of mortality, high rates of annual mortality, and with the plot that had the highest rate was 70%. So that's annual mortality. So that's 70%. That's less than a couple of years. You'll have complete stand death. But again, it varies dramatically. And one way that it varies, I just wanted to mention this, is that young stands seem to be much more resilient in the face of rapid Ōhiʻa death. And we don't quite understand why, but that certainly is a strong pattern. And we're looking into the different factors that could cause this difference. So this graph is showing the annual rates of mortality in stands where the average diameter of ohia trees is less than 15 centimeters in diameter. whereas the orange bars represent stands that have an average diameter greater than 15 centimeters diameter. And so you can see the rates of mortality are much higher in those older, bigger stands. That could be because there's more soil in those stands, could be that the young trees tend to not become wounded, which is an issue I'll get into in a second. Any number of factors, but as we're not quite sure about the mechanisms, we are certainly sure about the patterns. So young trees, like I said, are much more resilient in the face of Ceratocystis and its potential to kill trees than older trees and larger trees. This is really important to know and understand that Ceratocystis is characterized as a wound pathogen, and we've known this ever since we discovered Ceratocystis was the causal agent of Rapid Ōhiʻa Death. Ceratocystis is well known and well studied in other systems, other forest systems across the world, and it's widely acknowledged that Ceratocystis, including Ceratocystis lukuohia, needs to have a wound to enter the tree and kill it. If a tree is unwounded, no matter whether there is an abundant amount of Ceratocystis inoculum around it, that inoculum can't get into the tree and can't kill the tree. So that's a real Achilles heel of the fungal pathogen, that it needs a wound to get into a tree. And that has a lot of implications for how we think about managing the disease, which I'll get into in a little bit. Just want to really quickly go through this schematic of the disease cycle with you all, so you have a strong foundation, a good idea about how the pathogen actually kills ʻŌhiʻa trees and why, and also where those weak spots are in terms of the places where we can exploit in terms of management to protect trees, exploit the weaknesses of the fungus. So first in the upper left, we have a healthy, happy ʻŌhiʻa tree. At some point that tree becomes wounded, whether it could become wounded by a catastrophic wind event, like a hurricane or a heavy wind storm. It also could be wounded by things on the ground, such as feral ungulates, pigs in the schematic, or people, weed whackers, what have you. Anything that causes a wound makes the tree susceptible to infection. And if there is inoculum that's carried either by the wind or that's in the soil and is moved around by animals or people or machinery, if that inoculum gets into the tree will become infected, and it will start to produce, the fungus will start to spread within the tree. And you'll see that in the lower right hand portion, and then moving over to the left side of the lower left side of the diagram, the dead tree will start to become, or the dying tree in some cases, will start to become colonized by beetles, by ambrosia beetles. Those beetles will bore into the tree. They seem to really hone in on areas where the fungus is present. They will start to kick out boring dust, what's also called frass. They will kick that out from the tree. That is the source of inoculum, of infected material that is then transported from place to place, from that wounded tree to other trees, whether by wind, whether by animals, whether by people. That is the source of infection. So without the beetles boring into those trees and creating the frass in the first place, the tree would die, but the inoculum would just stay inside the tree, and would not pose a risk of infecting other trees. So as we move up through the diagram, we see the infective frass coming out of the tree and then providing the conditions when there are wounds of infecting more trees and so on around the cycle, as we see. So we have kind of two sources of two kind of places where we can stop infection. One is by stopping wounds, which is not possible to do when it comes to wind, but it is possible to do when it comes to ground wounding. And we can also try to stop the cycle by controlling beetle activity. And those are two primary management tools that we're developing as time goes by to protect trees from getting infected in the first place.
Excuse me, Flint.
Yes.
There's a quick question in
Oh,
the
great.
chat. Someone is asking if, well, regarding wounds, are beetles still a major vector, or are they secondary to mammal-caused wounds?
Oh, so we know from work that Kyle Roy, our Forest Service entomologist, that works with us, we do know that beetles are capable of carrying inoculum on their bodies and may carry it to other trees and infect trees that way. We think that while that occurs, we're not sure how often that occurs. The more important role that we know at least at this point, it seems that the more important role that the beetles play is creating the frass in the first place. And so the frass comes out of the tree and then other things move it around. Beetles may move it around, but it's probably more likely that other organisms are moving around or that the wind is moving it around, which is also the case. But the importance of those different kind of vectoring of those vectors is probably differs from place to place and situation to situation. But that's kind of the collection of vectors that we are aware of at this point. And again, sometimes humans might play a big role. Sometimes animals might play a big role. Sometimes wind might play a big role. And sometimes beetles might play a big role. So, but we just can't say, okay, this is the predominant vector system. Although in places we know that ungulates really seem to be a major vectoring agent. I hope that answers the question. I wish I could see, but I can't. Should I move on, Tristan?
Yep, that was great. Mahalo.
Okay, perfect. So how do we know that frass is important and carries the inoculum? We've done the work. Mark Hughes led a great research effort to understand the role of frass as inoculum and collected frass, tested it. Lo and behold, a large proportion of that frass that he collected out of infected trees contained inoculum that was viable and that produced spores that would, if they got into trees, would kill those trees. So we know that from work. We also know that when we're thinking about ungulates, we know that pigs and other feral ungulates carry Ceratocystis on their bodies, particularly on their hooves. And we know that from work that Kealoha Kinney and his research group have done in collaboration with hunting groups around the Big Island. Kealoha was in contact and worked with hunting groups staging tournaments across the Big Island, hunting tournaments, and as those tournaments proceeded, the hunters that would bring the animals back to a central location where Kealoha and his team would collect samples off of the animals, and lo and behold, a large proportion, about half of the animals that were sampled really at random across the island. They weren't hunted particularly in areas where Rapid Ōhiʻa Death was present. It was just wherever the hunters decided to hunt for those animals on that particular day. Half of those animals came back with carrying material that contains Ceratocystis huliohia or lukuohia or both. So that's pretty striking when you just have a random sample of feral pigs in the forest that half of them are carrying around Ceratocystis material. So we know that the ungulates carry it around in the forest. And just to recap, Ceratocystis is a wound pathogen. We know that beetles are producing frass in infected trees. We also, it's clear anybody who's been in the forest knows that ungulates move through the forest, and particularly where infected trees are present, and they have the potential to pick up and spread the inoculum. So the inoculum that could infect ohia trees. We also know that ungulates wound trees. Pigs in particular wound the roots of trees more often than they wound trunks of trees, and we're working to prove just over the course of the next couple months to prove that the inoculum can get into roots and is picked up by roots and can then migrate into the tree at large, into the trunks, and kill the tree. The one thing to note though is that wounds in ohia are short-lived. They don't last a whole lot of time. And the Ceratocystis needs a fresh wound to enter the tree. So we think that the wounds that allow for inoculum to enter the tree are open to infection for about a month, roughly speaking. So that window is not forever once a tree is wounded. It's actually a relatively short time. So the inoculum has to get into that tree at that time. So those are some factors when we're thinking about management that come into play in terms of dealing effectively with the disease in terms of protecting trees from it. So I want to show you some results from some of our work, and the two things I want to talk about are our inventory work, where we've been tracking the mortality of trees where Ceratocystis is present and has the potential to kill trees in a few of our study areas. We do that by setting up these monitoring plots. They're about a 1/4 acre in size. We go through the whole plot. We count the trees, we measure the trees, we note whether they're alive or dead. So this scheme shows time 0 when we establish the plot, noting, and the green blobs are living trees, the red blobs are dead trees. We'll go back a year later, and 2 years after that, and 3 years after that, just every year, noting how many trees are still alive and how many are dead. And we'll test those trees to see if they are positive for Ceratocystis or not. And so that gives us the ability to really track the disease and develop these annual rates of mortality values. We're also sampling for viable inoculum of Ceratocystis in soils around these stands and in these areas. And one of our studies is to look at the amount of viable inoculum in soils in areas where ungulates are abundant compared to areas where ungulates have been excluded, often fenced areas that are managed by either DOFA or by the national park or other land management institutions. And so this just shows a schematic of where and how we sample across the soils across a hectare sample plot. And then we bring those samples back, we process them so that we can then draw out and culture the Ceratocystis inoculum in the soils if it's present. And we do that with a carrot baiting technique. Ceratocystis happens to love to grow on carrots. And so if Ceratocystis is in soil, we can easily detect it with this carrot baiting technique, and then know, okay, we have this level of Ceratocystis presence across this sample area. And so I'll be talking a little bit about that in a second. So I just quickly want to get into some of the areas across the Big Island where we've been looking at rates of mortality and looking at some of the factors that affect mortality. And so this is Ka'u Forest Reserve and adjacent Kaiholena forest that's managed by the Nature Conservancy. The Nature Conservancy has been established a fence and has been keeping ungulates out of that fenced area for quite some time. That landscape is there on the left side of the screen. Again, the white and red dots indicate mortality, ʻŌhiʻa mortality, and there are plenty of ʻŌhiʻa trees inside the fenced area, but the levels of mortality are virtually zero. I think there's that one tree that was noted recently as dying. So you can see the effect of fencing in excluding Ceratocystis-induced mortality across these ʻŌhiʻa stands. It's quite dramatic outside the fence, virtually non-existent inside the fence. And our soil sampling kind of follows that pattern. The upper right image shows our sampling regime in the fenced area where pigs are excluded. The green dots signify soil samples, and the fact that they're green indicates that no Ceratocystis inoculum was found at that point in the forest. Now, when you compare that with the Ka'u Forest Reserve next door, adjacent to the fenced area, so it's an unfenced area, pigs are abundant, they're running around, there's a lot of mortality of the trees, and frass is coming out of those trees. So the potential for further wounding is quite high, for infection is quite high. And what we see in the soil sampling is that a large percentage of those soil samples were positive for Ceratocystis. So we're finding that inoculum in those particular soils across that landscape. So it's not really too surprising that we're seeing those increased levels of mortality, because the inoculum is there, it's available for ungulates to pick up, and then as they wound trees, it's easy for those trees to become infected with the disease. This is another study area up in Ola'a, in the national park, and in the forest reserve, adjacent forest reserve, the national park area. The small tract unit is fenced. It has been fenced for a long time. Ungulate ingress has been tightly controlled, and so it's effectively ungulate free. So again, the green denotes no positive, no positive present, or no presence of Ceratocystis in the soil, Ceratocystis inoculum. And in the unfenced area, you see, again, lots of ungulates, lots of mortality, lots of inoculum in the soils. 56% of those samples came back positive. An adjacent forest stand that we've been monitoring for multiple years, you can see the levels of mortality are quite high, and that's an image of all the dead trees across that stand. This is very near the volcano waste transfer station. So what we're seeing is 50% of the ʻŌhiʻa were dying within the 4 years that we were sampling, that we were monitoring the plot, and we continue to monitor the plot. But initially we had close to 200 trees across the stand. Now we have less than 100. So dramatic mortality associated with Ceratocystis in these sites. The Mauna Kea forest up high, a similar situation. We don't have a fenced control in this area, but we were curious about the levels of Ceratocystis in the soils associated with what we're seeing is high mortality area. We didn't see quite such high levels of positivity in the soils, which is interesting. We don't have any beetles in this area producing frass, so that might explain why we have less Ceratocystis inoculum in the soils. But really what I wanted to point out is the dramatic mortality that we're seeing in this area. Greater than 60% of the trees died within 2 years' time. So just want to emphasize the dramatic effect that Ceratocystis can have in areas where it really takes hold and is killing trees. In Lapahoehoe, moving on to Lapahoehoe, similar situation. This is a zoom in of one of the areas of the Valley of the Giants, where I showed the patterns of mortality earlier. In the unfenced areas, and I'll get back to this fenced area in a second, that we're showing across the landscape. This yellow area on the far left side is a fenced area, a small fenced unit where ungulates have been excluded for quite some time. And you can see that we've got a lot of mortality around that fence unit and very little inside. Outside the unit, we found in our soil sampling, we found about 20% positive. 20% of the samples that we collected came back positive. So not super high compared to some of the others, but again, it's associated with dramatic mortality. Greater than 50% of the trees in our monitoring stand in that area were dead within 2 years. So again, dramatic mortality associated with the presence of Ceratocystis across this landscape, the recent presence of Ceratocystis. That's just a zoomed in image of that fence, of that small fence unit, showing the dead trees around it and very few dead trees inside the unit. So here's a little bit of math for you. I know math can be hard for people. 5 out of 4 people struggle with math. I know that for a fact. But this is our little formula. If you've got Ceratocystis lukuohia and you've got beetle frass and you've got feral ungulates moving the frass around and wounding trees, you're gonna see a lot of dead ohia. Can we protect ohia trees from rapid Ōhiʻa death? Definitely, we certainly can. And the main way we can do that is by keeping and controlling feral ungulates from wounding trees and infecting them with Ceratocystis that those ungulates would otherwise pick up from soils. So if we can keep ungulates away from ohia trees, stands, and forests, they will be much more likely to persist in the face of Ceratocystis-induced rapid Ōhiʻa death. And that's all I wanted to say. I'm sorry it took me so long to tell you all that, but I'm happy to entertain any questions that you have now. And I think I can just escape and stop sharing.
Thank you very much, Flint. There is another question that just entered the chat. Well, first, actually, JB Friday made a little note, which is kind of reiterating some of the things that you said, that, you know, the side-by-side maps of fenced versus unfenced areas, with fenced areas having much lower rates of disease, indicates that fences make a big difference. As you said, fences keep out goats, pigs, sheep, and cattle, but not beetles. Of course, windstorms don't respect fences. There are many ways to get trees wounded. And then Geraldo just asked, are there environmental limits on ambrosia beetles, or are they virtually everywhere we have ʻŌhiʻa?
Yeah, well, first, just back to JB real quick. I definitely, obviously, trees can become wounded during wind events, and wind events can certainly have the potential to carry inoculum if it's available to be picked up, which we think it is in certain cases. That can happen in a punctuated sense, but we cannot control that. But I think what we're seeing is this, what we think of as kind of chronic wounding in the areas where ungulates are active. So it's not a punctuated event. If ungulates are there, they're wounding trees on a daily basis. And to the extent that inoculum is there, then those trees are highly likely to become infected. And so we kind of think about those two types of wounding events as different beasts. One that's ongoing seemingly all the time where ungulates are present, and the other is, you know, an event basis that could occur quite frequently, but it's a single event. And so that's, again, you can't control that, and we think that tends to be the mechanism by which new outbreak areas where we hadn't seen Ceratocystis infection and mortality before, we think that's how those new outbreak areas become established, and through those wind events more likely than that chronic wounding event. So I'm sorry, Geraldo had a question that I now have forgotten.
No problem. Yes, so his was, are there any environmental limits on ambrosia beetles, or are they virtually everywhere we have ʻŌhiʻa?
Yeah, great question. Great question. There are, and ambrosia beetles do have an elevation and temperature limit, seemingly. I just mentioned it really quick in the talk that one area that was fairly high up, plus certainly higher than 4,500 feet, the ambrosia beetles weren't present. So they definitely have an elevation limit in terms of both their presence and their activity. And that's what we've seen. I think I don't feel completely confident giving a single elevational figure, but it's probably around 3,500, something like that. Kyle Roy and Bob Peck have done a lot of work with the beetle activity and patterns of abundance, and they could speak much better about that than I could. But yes, there is an environment. There is a temperature-induced limit, which, yeah, and that clearly plays into disease transfer and infection and mortality. Absolutely.
And then one more from Geraldo. Do we know how long inoculum is viable in soil?
We don't yet know that, and so that's a research question that we hope to tackle, but we think that it can be fairly long-lived, you know, months to maybe years, something like that. I'm not sure if anybody else is on the meeting that could speak more to that, but that's my understanding, that it's the inoculum has a capacity to persist in the soil for quite some time.
It's incredibly frustrating. Thank you. One more, or actually two more. One from Phil. He's asking if dead trees inside of fenced areas were killed by rot or other causes. And I actually had the same question because I noticed you had them listed as suspects. So I wasn't sure if that tested any of them. Yeah,
so that's a great question too. unless we test a tree, so go out and sample the wood, bring it back, look to see if there's Ceratocystis DNA contained within that wood sample, we can't really say that we're 100% positive that that particular tree was killed by Ceratocystis. But once we start to sample a lot of trees that have died at the same time, the same kind of episode, and those are turning up positive, we're pretty confident that the trees around them were also positive for Ceratocystis. But you really can't say 100% guarantee that tree died from Ceratocystis unless you've tested it.
Awesome, thank you. And then one more, with the frequency and severity of storms expected to increase with climate change, including wind, can we expect a greater spread of the Rapid Ōhiʻa Death in the future due to this?
I would expect that to be the case, unfortunately. Yeah, that, you know, if we continue to have inoculum in the form of frass present and capable of being dispersed during wind events, and those wind events increase in frequency, I would unfortunately suspect that those kinds of mortality inducing events would increase. Yeah. The other thing is, since we're talking about climate change induced effects, increasing temperatures are another factor which may increase the elevation, increase the effective habitat for beetles. So as things warm, beetles may be able to move higher up on the island and become more active, create more frass, etc. So that's another climate-induced factor.
Mahalo for touching on that, because that was kind of actually going to be my segue, was the beetles, and yeah, of course, climate change is no bueno. So thank you so much for answering all those questions. If anyone else has any more questions, feel free to ask or drop them in the chat with the couple minutes we have left scheduled for this talk. And I'm going to take a second to drop in that Google form link in the chat. So if you folks do have any feedback, we really do appreciate that. And there is a place at the bottom of that to be able to sign up for our mailing list. And I believe we do have Kyle Roy scheduled later on in our webinar series. So at least she can touch on a couple of things related to Flint's talk.
Oh yeah, excellent. I'm glad she can correct all the things that I got wrong.
Okay, Tom has a quick question, and he's asking if you could briefly talk about restoration and reforestation efforts.
Oh, yeah, good question. Well, so the good news is, as I said, young trees are able to persist in the face of the presence of Ceratocystis inoculum, which is great. So planting young trees is a good thing, even if you're planting them in areas where the more mature trees have succumbed to the disease, replanting is great. Also, and this is something I absolutely did not touch on, there's a very active research program looking into disease-resistant ʻŌhiʻa strains and varieties and species, and that is ongoing here at the Forest Service and with collaboration. And Mark Hughes and Christian Giardina and others have been leading that up, and they've already started to perceive resistance in certain populations of ʻŌhiʻa. And so we hope to continue with that work and continue to develop and and outplant strains of ʻŌhiʻa that are varieties of ʻŌhiʻa that are resistant to the disease.
That is very exciting and promising. One more question from Pamela. She wants to know, is there a difference between wounds from pigs to roots versus other ungulates on bark in inoculation and mortality?
I don't think we know that yet. I think any wounds where the inoculum can get to, as long as they're fresh, are going to allow for infection and eventual mortality. We've been less sure about the role of roots in carrying the pathogen, but as we've been working on that issue, it's becoming a little bit more clear that wounds are a viable point of infection, and whether our next step is to demonstrate that those root infection points lead to infection throughout the rest of the tree. And so that's kind of the next step that we're just kind of starting right now, actually.
Wonderful. Thank you so much, Flint. That was very, very thorough. You know, I think if there is any more questions, and you can drop them, but I think we're going to wrap up. And really appreciate you folks participating. This was a great start to our 2023 series. So mahalo everyone for attending. This video is going to be uploaded to YouTube, and we'll be sending out a follow-up email to all of you folks who attended. So yeah, if you guys do want to connect with Flint and you need that information, I'm sure we'll be happy to forward his email at the very least.
Yeah, great. Yeah, please reach out if you have any follow-up questions.
All right, mahalo, Flint, ahuiho. Thank you everyone for attending, and you guys have a great rest of your week. Aloha.
Great. Take care. Bye-bye.